help out with this comparison between the two, I'm going to pull up the lectures again. And I'm just going to jump here real quick. This is the end of both aerobic and anaerobic respiration. Respiration refers specifically, when I think of respiration, I think Krebs cycle and electron transport chain are going to happen. But, and that's absolutely true. Okay, so if you see respiration, two of the pathways that are going to be involved are Krebs cycle and then electron transport chain and oxidative phosphorylation. If we're talking about glucose being broken down, glycolysis has to be involved as well. Because what's the first part of the Krebs cycle? So what's the first step? And I'll back up to that. Yeah, breaking down pyruvate. And where does that come from? That comes from glycolysis. Okay? So in order to get to the start of the Krebs cycle, we have to make or yield pyruvate somehow. There are multiple ways, but we oftentimes, and in here, we're pretty much exclusively talking about breakdown of glucose or some other monosaccharide. So one thing that both of those pathways have, or that aerobic respiration and anaerobic respiration have in common, is that they share glycolysis, Krebs cycle, and electron transport chain. What's different? And a lot of people got this. This part was really good. So what's different between them?
The amount of ATP.
That's one. The amount of ATP differs. We defined that in prokaryotes, bacteria. Typically, we get about 38 ATP from aerobic respiration. Anaerobic respiration gets more than that, or I'm sorry, gets less than that, but it gets more than we'll find in fermentation. Why do we get less ATP out of anaerobic respiration?
Does it have to do with the oxygen not being the final?
Absolutely does. Absolutely does. So let's go back to this chart, again real quick. So one of the things that we said, oxygen, out of all those final electron acceptors, oxygen is the most electronegative. Therefore, those high energy electrons from NADH go into the electron transport chain, and they can release the most energy over the course of this electron transport chain if oxygen is the final electron acceptor. As they're releasing that energy, they're driving those hydrogen ions across. So the more energy released leads to more hydrogen ions being pumped to the periplasm, which ultimately leads to more ATP yield through oxidative phosphorylation. In the case of something like nitrate reductase, which is one of the complexes in anaerobic respiration, one of the things that we said happens is that these last couple of complexes aren't used, therefore there's not going to be electrons flowing through them, therefore there's less hydrogen ions that are going to be pumped across. In fact, the nitrate reductase replaces this complex right here, and in doing so, since the electrons aren't losing as much energy, since that energy isn't being used to pump the hydrogen ions across the membrane, there are fewer hydrogens to flow through the ATP synthase, therefore leading to overall less energy. How much? I don't know. I really don't, because it differs for each of those final electron acceptors. Out of the three of them, nitrate yields the most, sulfate yields the second most, and carbonate the third. I usually tell folks, define this number as being somewhere between 4 and 38 total ATP yielded. And this number comes from, I should define where this comes from. Glycolysis gives us how many? 2. Krebs cycle gives us how many more? 2 more. And then from oxidative phosphorylation, at least when oxygen is the final electron acceptor, there's 34 more for bacteria. That's where the 38 come from. So that's why I say for anaerobic respiration, it's somewhere between, it's more than 4, but less than 38. Okay? What products get produced in aerobic respiration? So what kind of products do we find throughout the pathways being formed? And I define this a little ambiguously, but oxygen gets reduced to form what? Water. Yeah, so H2O gets formed. What else forms throughout the process? CO2 does as well. The full oxidation of carbon, or the complete oxidation of those carbon molecules from the glucose, produce the CO2. When do those get produced? What glycolysis, Krebs cycle, or electron transport? Yeah, Krebs is where that CO2 gets produced. Good. What about and those are kind of when we think of the basic equation that we correlate with respiration. This is the typical equation: C6H12 O6, and we get out 6 H2Os, and we get out 6 CO2s. There's something else in here I'm missing. There's O2 over here also. But there's more that happens. This is such a simplistic explanation of what's going on here, right? So what else? What coenzymes get reduced in this process? So like in Krebs cycle, what are the major redox reactions? Good. That's so NAD plus to NADH, and what's the other coenzyme? FAD to FADH2. So those get reduced at some point, but then they get oxidized within the electron transport chain, right? So we see redox reactions there. Because there's no change of them throughout the whole process, if we were to look at it as a single thing, that's why they don't get included in this basic equation. But there are many small steps that are happening between this first step here and this last step over here. Some of those are those redox reactions. Okay. Otherwise, Abby, go ahead.
Would you count those as products?
Yeah, so that's why I looked at it and I was like, that's kind of an ambiguous question. I would say that they are, that, I guess what I was trying to get at with that question is more like what's actually getting produced throughout this whole process. If you were to look at like each of the individual steps, what's going on? And so if you included them in there, that's perfectly fine. I would just say, but they are oxidized to NAD and FAD at the end of the process. Okay, so there's a little caveat to it. What about anaerobic respiration? In anaerobic respiration, what gets produced? So nitrate gets reduced to what when it's a final electron acceptor? Nitrite or nitrogen gas, right? Sulfate gets reduced to hydrogen sulfide. Carbonate gets reduced to water and methane, CH4. Anybody been to a dump? I have many times. So these days dumps harvest energy in the form of what? Methane. If you ever drive by a dump at night, you might see large blue cones of fire coming out of the dumps. That's the excess methane gas that's being released. That methane is all produced by bacteria that are breaking down carbonate in those dumps. Anaerobic respiration. And of course, again, we'll see, because the pathways are identical otherwise, we see NADH being produced and then again oxidized, and we see FADH2 being produced and then oxidized in anaerobic respiration as well. Okay? What kinds of phosphorylation occur? That's kind of the last thing we haven't covered here. But what kinds of phosphorylation occur in both of these forms of respiration? And that's the one behind, right? Oxidative phosphorylation. What's that?
That's an anaerobic too.
Oh, yeah. Yeah. So that's one misconception that I think is here, because oxidative is in the name. We would assume oxygen, right? But the oxidative here doesn't refer to the oxygen. It actually refers to the redox reactions that occur that yield that ATP. So what really it's meaning is the full oxidation of carbon, or the oxidation of that NADH to NAD, or FADH2 to FAD, that leads to the production of that ATP. Yeah. So that's one. What's the other type that we find? We see substrate level in glycolysis in Krebs. Okay. Yes, yes. Does this help? Does this help clarify some issues? Yes, yes. So the biggest things that are different between aerobic and anaerobic really are the final electron acceptor. That first and foremost is probably the biggest difference. And that final electron acceptor then affects the yield of ATP. It also affects the final products of the reaction as well. Questions on these?
So what is the final electron acceptor for anaerobic respiration?
So depending on the organism, it can be carbonate, CO3 2- or negative, sulfate, or nitrate are the three major ones. Any other questions? Thanks for asking that question. So I forgot to cover that. So the oxidation this question is the complete or incomplete oxidation of carbon. I guess I should have defined this as the complete or incomplete oxidation of carbon from glucose. That's what we're really referring to here, is those 6 carbons that are in the original glucose molecule, are they fully oxidized? And the full oxidated form of carbon is CO2. When do we produce CO2? Krebs cycle, TCA. When do both of those occur for aerobic and anaerobic? Yep. So we see complete oxidation of CO2 in each of those. In other words, one of the ways to think about this is in aerobic and anaerobic, we're harvesting as much of the energy as we can from those high energy electrons that are found in those carbon bonds in the glucose molecule. Yeah, thanks for asking. I forgot that. Erica.
So what are the metabolic pathways utilized in anaerobic?
Same ones as aerobic. Same ones as aerobic. Glycolysis, Krebs. Other questions?
Does H2O get produced in anaerobic too?
It can in some forms of anaerobic. So, for example, like I'm pretty sure carbonate, sulfate, I think water gets produced. It's just dependent on the chemistry of how the reduction of that final electron acceptor occurs. Shall we move on to fermentation? If there is one metabolic pathway that should be the state metabolic pathway of Wisconsin, I argue it needs to be fermentation. What are we known for? Beer and cheese. In other words, alcoholic fermentation and lactic acid fermentation. Okay? I think we should fight for this one. In fermentation, what's the major of the three pathways that we've talked about? Krebs, electron transport, glycolysis. Which is the only one that we find? Perfect. Great start. Glycolysis is the only one. I'm going to go back to an image of glycolysis because this really is the heart of fermentation. Since glycolysis is the only one, how many ATP get produced? 2. Great. 2 per glucose. In other words, cells that perform fermentation are not tapping in to the energy that they could from glucose. Okay? Really what happens at the end of glycolysis with this pyruvate is it's kind of like a decision point for the cell. And what I mean by that decision point is whether it performs aerobic respiration, anaerobic respiration, or fermentation is based on the environment. So for example, if oxygen is absent, obviously you cannot perform aerobic respiration. If the certain electron acceptors, final electron acceptors, aren't present that you need. So like for example, methanogens are anaerobic microorganisms. They perform anaerobic metabolism where carbonate is the final electron acceptor and they produce methane. If carbonate's not available, they have to perform fermentation to generate energy. So it's dependent on what's there. It's also dependent on the genes that an organism has, and kind of you could think of it as the default settings. For example, in the presence of glucose, yeast, even if oxygen is present, they're going to perform fermentation first. Fermentation actually, out of the three pathways, leads to the quickest growth in the shortest period of time. The downside of it, you burn up your available carbon source really, really fast. And so what we find is that cells that perform fermentation oftentimes will undergo a quick growth phase. So let's let this here represent our growth. And then they'll kind of level out for a while in terms of numbers, and then they switch to a different form of metabolism, anaerobic or aerobic respiration, to use the byproducts of that fermentation. And when they do, you'll see a second growth phase, a little bit slower. This is really not important for the exam. This is something we'll come back to down the road, probably like 5 weeks from now. Okay, but just wanted to point that out. In terms of fermentation, we get 2 ATP, and in glycolysis, we also get 2 NADH, right? In the ways that we've talked about so far, how is NADH oxidized back to NAD+? When does that occur? Electron transport chain. Perfect. What if a cell doesn't use the electron transport chain to oxidize that NADH? Well, if we keep reducing NAD to NADH, eventually there's not going to be any more NAD. And if there's no NAD, can glycolysis occur? No. The purpose of fermentation is to oxidize NAD, is to oxidize NADH into NAD so that glycolysis can continue. That is the main function of fermentation. It's not as if yeast have said, let's go have a party. It's not as if the lactic acid bacteria have said, let's add some cheese to this party, or yogurt. Sole purpose is to oxidize or to produce that NAD so that cells can continue to yield energy through this process of glycolysis. What is the final electron acceptor? Yeah, most books will list them as organic molecules. Usually the first one in this step is the pyruvate that's produced at the end of glycolysis. That pyruvate will ultimately be reduced into one of a number of acids, gases, or ethanol. We're just going to focus primarily on two of them: alcoholic fermentation, lactic acid fermentation. There are other forms as well that produce other byproducts: succinic acid, acetic acid, vinegar, formic acid, propionic acid. There are many different forms, and each of those is just going to be different in different sets of microorganisms. Oh, since we're only Abby, go ahead.
So if you were to ask us what the final electronic center is in fermentation, what would you?
I would say pyruvate. Yeah, typically that's what we teach folks, is pyruvate is that primary one. We are not going to go through in depth the chemistry to this process. Of course, not working. But what I want to just kind of focus on is just these two steps briefly to give you an idea of what's happening. So the top part here is representing glycolysis. It's showing the NADH that gets produced. It's showing the ATP that gets formed. And then at this point, what this image is really trying to show is that, hey, look, here are those redox reactions. Pyruvate will get reduced to form lactic acid. What kind of enzyme is involved? Somebody said it. Somebody said it. I heard it around this area. Thank you. Name lactic acid dehydrogenase, LDH. On the right-hand side of this image, first there's going to be an oxidation of carbon to form a 2-carbon molecule, acetaldehyde. That gets reduced to form our ethanol, and that's where we get the gas and the alcohol in alcoholic fermentation. Now, you've seen in some of the phenol red tests that you looked at yesterday, there were gases produced. Organisms like E. coli actually perform a number of different fermentations. They can form lactic acid. They can form acetic acid, which is a 2-carbon molecule. In those cases where we get different types of fermentation, some of them can result in different gas production as well. It doesn't have to be CO2, could be H2, for example, hydrogen gas. Are the carbons in these molecules then fully oxidized by the time that we've reached the end of fermentation? So is the carbon oxidation complete or incomplete? Yeah, it's incomplete. I like to think of it as there's still some meat on the bone here. There is still some energy to be harvested from those products. And what we find is that some organisms can actually take that lactic acid, oxidize it back to pyruvate, and then use that pyruvate in the Krebs cycle. Human anatomy and physiology for the day. I know there's a number of people who play sports in here. I once upon a time did. Nowadays, just walking builds up the lactic acid. Okay? What happens to that lactic acid? Anybody know? Austin? It pools and you cramp, and what happens after that? Well, yeah, but what do our bodies do with that lactic acid? They oxidize it back to pyruvate. And they chew off the rest of the meat on the bone. They take that pyruvate, they put it through Krebs cycle. And from Krebs cycle, it goes to the electron transport chain. We get as much energy from it as we can. But when we do that, it's going to be dependent on oxygen being present, because that lactic acid is produced in times of quick bursts of energy when we need to produce lots of ATP in a short period of time. Electron transport chain, Krebs cycle, take a little bit longer to do. You get more energy overall from it, but it takes a longer period of time to do. And so for us, the lactic acid will eventually get oxidized and then broken down further. So some organisms do that. They will change metabolism in order to break down some of these byproducts. pH obviously drops, okay? If you listen to the lecture, this is Jake's science experiment from like 2 years ago. We made sauerkraut at home. He apparently liked it. I think it's disgusting. Red cabbage has an awesome natural pH indicator in it. So it's when you blend it up, it's like a purplish color. When it becomes acidic, it goes to a pink color. More basic, it kind of becomes blue almost. So that's one way you can tell that you actually have created good sauerkraut or that it's been fermented, is by that color change. And this one, obviously, drop in pH here. What's being formed? What's likely being formed when we make sauerkraut? Lactic acid. Yeah, that's the major one. Absolutely. So there are some bacteria that eventually populate sauerkraut that lead to the formation of that lactic acid. Great follow-up question. Would this organism be MR positive or VP positive in the sauerkraut? Ah, very good. Why? Good. Perfect. Below that certain pH level. So what can we say about the acids that are being produced? Stable or unstable? Stable acids. Lactic acid, acetic acid are two of the stable types of byproducts that get produced. Perfect. This here is, I'm going to blow this one up a little bit. This is just showing you guys some of the examples of what's produced. I'm not asking you to memorize these by any means. Again, lactic acid, alcoholic fermentation, those are our state, so we will stick to those. But here are some other ones that are formed. Some bacteria exclusively form one type of product. Lactobacillus, some bacillus species, and some streptococci produce only lactic acid. They don't produce any gas in the process. We call those types of organisms homofermenters. They form a single product, as opposed to heterofermenters, which will form a mixture of products based on the type of metabolism that they have. E. coli and salmonella are great examples where they can perform ethanol, or they can produce ethanol in some cases. Lactic acid, succinic acid, acetic acid, carbon dioxide, and hydrogen gas. So that is an example of a heterofermenter. Any questions with those basic pieces? What have we missed on the table here for fermentation? Are we missing anything right now?
I just had a question. If lactic acid is the final product, why isn't that considered the final electron acceptor?
Do we consider water to be the final electron acceptor? No. That's where I draw the comparison. It's like we call oxygen the final electron acceptor. It gets reduced to water. In the same way pyruvate gets reduced to lactic acid.
Okay.
Other questions? Devin?
The type of phosphorylation.
Oh, type of phosphorylation that's occurring here? Oh, substrate level. Substrate. Because, again, let's mechanistically link oxidative to electron transport chain. And we've already defined that in glycolysis, only substrate level phosphorylation occurs. Transfer of a phosphate from a couple of different molecules onto ADP to form ATP. So it's going to be substrate level. Anything else we've missed on it?
So it doesn't have complete oxidation?
Not complete oxidation, yeah. Because again, those carbons that come from the glucose molecule, again, the fully oxidized form of carbon is CO2. Well, here are 3 of those carbons in the lactic acid fermentation. They're in 2 molecules of lactate. And they've still got hydrogens associated with them, so these are, some of them are still reduced beyond their full extent. Same thing with ethanol as well. We've still got some carbon-hydrogen bonds, which indicates that there's some, that there is still reduced forms of carbon there. Anything else? Okay, so let me explain to you what your homework was going to be for Monday, okay? And then I got to come up with something new now. So, the homework was going to be to read a paper, a scientific paper about a bacteria that takes PCBs and breaks them down. Anybody know about PCBs? Who's from Appleton, Green Bay? PCBs are in the Fox River. So like they've been doing a lot of dredging over the past few years. They're a product of paper production, basically. And there have been found bacteria that break those down, so they're using them as alternatives. We were going to talk about an alternative form of metabolism, something we haven't covered. I don't think we're ready for that, and that's okay. We don't have to be ready for it. What I want to do on Monday then is I want to review these 3 topics, but I want to review them in the context of metabolic tests. And I want to bring in one additional metabolic test that we've talked about as well. So the ones that we're going to talk about on Monday, and these ones will be fair game for the exam. The phenol red tests, glucose, lactose, sucrose. I'd like to talk about MRVP again. We'll probably focus primarily on MR of the 2 of them, but I'd like to talk about that. I have this inkling that oxidase may be on the exam because that has to do with aerobic respiration, so we may cover that briefly. What am I missing? I don't want to worry about citrate. I wanted to do the indole test as well. I wanted to talk about tryptophan breakdown because what we have done so far, our blinders are on glucose, glucose, glucose as the only source of carbon. That's not true. There are others. Tryptophan is one of them. And so we'll talk a little bit about what if glucose isn't the carbohydrate? What if it's something like tryptophan that gets broken down into indole and pyruvate? Then what happens? How does it have to be metabolized? How does that dictate what form of these metabolic pathways, aerobic, anaerobic, respiration, or fermentation, how does it dictate which one will occur? So those are the ones that I'd like to focus on for Monday. I will come up with some, actually, let's do this. No assignment? No assignment. Study. Study. Okay? That's your assignment for the weekend. All right. Any last minute things that we need to discuss? All right, so keep in mind the 3 differences between those, or the metabolic pathway differences there. Otherwise, have a great week. And, oh, I do have one last announcement. Sorry.